Hey, so now we're doing the phosphate experiment in Chem 100A, and we'll be analyzing the phosphate concentration in low nutrient and contaminated seawater using the method of UV visible spectrophotometry. So phosphate, why is it important? It is present in uh, making life, essentially. The phosphate groups are present in DNA and proteins and all sorts of things. And so that makes it very important for life. Since it is necessary for life, that means, for example, when we want to grow crops, our plants need phosphate in order to grow well. So phosphate is a common additive in fertilizers. And something to keep in mind with that as we're using fertilizers all throughout agriculture is that the runoff from our agriculture then has a lot of this nutrient left over, and if it enters any stagnant water areas, a phenomenon called eutrophication can occur. Eutrophication. This is what happens when there's all this excess nutrient available in the water. All of a sudden algae can grow super well, and it grows so fast and uses up all of the oxygen available in this water, which causes all other life forms that rely on that oxygen heavily to die. Okay, so eutrophication can be a problem as a result of us using phosphate fertilizers. And one other note is related to the idea that phosphate is important for life, that makes this a non-conservative element in the ocean. Okay, all of the elements we have analyzed in seawater so far have been conservative elements, meaning their concentrations are roughly the same all the way through the depths of the ocean. But now phosphate has varying concentrations at different depths because all the sea life is using it, especially at the top of the ocean. Okay, and we are going to analyze this phosphate with UV-Vis. But there's some things we need to think about first. So first of all, phosphate is naturally colorless in solution. So we're going to need to convert it into some form that absorbs in the UV visible spectrum. Okay, and there are a couple different types of compounds that typically absorb in the UV-Vis. First of all, we're looking at electronic transitions. So this is very similar to the atomic absorption pre-lab lecture that I gave before, where we're looking at electronic transitions. But in the UV visible, we're usually more interested in molecules, and the types of molecules that absorb in this range tend to be compounds that have double bonds or metal coordination complexes. And in this experiment, we're looking at making a metal coordination complex. Okay, so we're gonna be doing a reaction in this experiment where we're converting our phosphate into a metal coordination complex using molybdenum as the center metal. So here's how those reactions are going to go. Taking our phosphate, and in acidic solution we'll react it with this molybdenum. Okay, and that reacts to form this product, which then we reduce further to form our coordination complex. So we take that product and we're going to react it with a reductant to form our colored product. Okay. So this product is blue. And just one thing I do want to point out, this 6 and 5, that represents the oxidation state of these different molybdenum atoms. Okay. Also, this reductant is composed of ascorbic acid and an antimony. Okay. So that's what we'll be doing first. We need to convert any phosphate in our solutions into this blue form, because a colored compound, that means it can absorb in the visible range. Okay, so we have talked a little bit about spectroscopy before, but that was in relation to atomic absorption spectroscopy. So that was where we did, we were looking at atoms. Okay, so atomic means we have just an atom like this, and an atom is able to undergo electronic transitions. Right, we can excite an electron, but since it's a sphere, we can't undergo any vibrations. There's nothing to vibrate, and there's also, you can't rotate something that's a sphere because it's just still a sphere. On the other hand, however, with molecules, if we do molecular spectroscopy, I have something, simplified molecule here, 2 atoms connected by a bond. Once again, we can do electronic transitions. But now also we can do vibrational. That bond can get longer and shorter. And we can also do rotational, spin this whole thing around, right? Electronic, vibrational, rotational. So what does this mean? In atomic spectroscopy, you might remember we used a hollow cathode lamp as our light source in order to emit just a single wavelength, because we knew that our atoms would absorb right at that single wavelength. This is called a line spectra. Okay, the hollow cathode lamp is called a line source, and our absorption spectrum would just be at a single wavelength. However, with the molecules, now that we have these vibrations and rotations happening, our peaks get broadened. Okay, that means that these molecules aren't going to absorb at one very, very specific wavelength, but it'll absorb more over a broad set of wavelengths. So that's going to lead to some differences between our UV visible spectrophotometer and that of the AA. Although they're very similar, there are some different things we're taking into account now. Okay, so let's look at what this instrument looks like. UV-Vis spec. I do want to note that this is going to be oversimplified. And also I am drawing out what a single beam UV-Vis spec would look like, but double beam, and that's what your instrument in lab looks like, but double beam spectrophotometers are also commonly produced. Okay, so first things first, kind of like I've touched on a bit, is our light source. Okay. For UV visible spectrophotometry, we want to be able to emit in the UV and the visible. So to emit, in the UV range, what's commonly used is a deuterium lamp. Okay, and in the visible range, you use an incandescent light lamp of some sort, including like a classic tungsten light bulb, kind of like the one I drew. Okay, incandescent. And note that the light from this kind of light source is over a broad range of wavelengths and just emits in all directions. So up next, typically in a UV-Vis spec, is a monochromator, and I'm going to break down the monochromator again in an oversimplified way here. But a monochromator, the whole purpose is to select just a single wavelength. So we have this lamp that's emitting over a range of wavelengths over the entire UV visible spectrum, but we want to just select one of those and send one wavelength at a time through our sample and see how it absorbs. Okay, so we're gonna, first things in this monochromator is a lens, specifically a collimating lens, which means it's taking this light that's going in all directions and it's going to make it go parallel. Okay. Once your light is going parallel, next up is a prism or a grating. Gratings are more commonly used, but prisms make it kind of easy to understand. We'll draw a prism here, and what the prism and grating does is break your white light over this whole range of wavelengths into all the constituent wavelengths that are inside. Right? So if we have visible light, it's going to break it down into all of those colors. Right? Okay. Up next, we want to just pick out one of those wavelengths. So next is a slit, which makes it so that all of the other wavelengths, apart from the one that we want to select out, are blocked. And like in this example, looks like the yellow would go right through. Okay, so typically, when we want to select one wavelength, you could theoretically move that slit around in order to, just move that up and down, and you would get a different color coming through. But in practice, it's actually a lot easier to move the prism or the grating itself in order to select the wavelength. So that's what happens when you use your UV-Vis spectrophotometer and you ask it to select a certain wavelength. The grating moves into the right position to get that specific wavelength out of your lamp. Okay, so here we are. We have a specific wavelength of our lamp with some intensity I0, and we send that through our sample. Let me just specify here, go back real quick. All of this is a monochromator. Composed of a collimating lens, a prism or a grating, and lastly a slit. Okay, and then we have our sample. Which, once our sample with some concentration of colored compound in there, it's going to absorb some of that UV visible light and causes the intensity to decrease. Um, and the transmitted light has an intensity I, which we detect on our detector. And the detector is typically a photomultiplier tube. Photomultiplier tube essentially takes a photon that hits a plate that emits electrons, a diode that emits electrons, and that electron goes and hits another plate, which then emits another electron. So now you have 2 electrons, which hit another plate, which emits another electron. Now you have 3, 4, 5, 6, and so on until you have so many electrons that you're generating a large enough current that you can detect really well. Okay, so notice one other difference here between our UV visible spectrophotometer and that of the AA is that this monochromator comes before the sample this time. Remember in the AA, we had our hollow cathode lamp light go straight into the sample through the flame, and then we had a monochromator to get rid of any noise or other wavelengths that we don't care about. Now we need the monochromator before the sample so that we can select the wavelength we want to send through. Okay, so specific to this experiment, some things you do. First of all, you're making this complex, but you don't know exactly what wavelength it absorbs at best. You want to be able to do your measurements at the selected wavelength where you're getting the best absorbance. So the first step you do is to determine this lambda max, the wavelength where you absorb the most. So you're going to scan across all the wavelengths. Okay, and that's again happening by moving the grating inside the instrument, and you'll find the peak. So it'll look something like this, where you measure across all of the different wavelengths that your light source is able to emit and look at the absorbance, and it might look something like this, and you'll notice that's the peak. So what wavelength does that occur at? And you want to do all of your measurements from here on out at that wavelength because that's where you'll get the best response from your instrument. Next, you'll, in this experiment, again, be converting your phosphate into this complex that's blue, and it might be helpful to know how long it takes that reaction to go to completion so that you're not putting your sample in too early. So this is called the time determination step. So you'll want to set your instrument to the lambda max and measure just at that wavelength. Okay, and you're gonna add your reagents right away. Add your reagents and start measuring the absorbance immediately. Okay, so what this is going to allow you to do is to make a plot that looks something like this, where you have time on the x-axis and the absorbance on the y-axis. At the very beginning, when you first add your reagents, your solution will still be clear for the most part. So your absorbance will be very low. Might look something like that. But as time goes on, this reaction is going forward and you're forming more and more of that blue colored complex. So your absorbance is going to increase. But eventually, you'll have reached completion of this reaction and you'll level off to an asymptote. Okay, so the time determination you're looking for is the shortest amount of time it takes for you to form that complex in completion. So in this example, it looks like it's something around here. right? Because after that amount of time, you're not forming any more of your complex. Okay, once again, we're doing an external calibration method for this experiment. But we are expanding on it just a little bit this time by making what's called a working curve. Okay, external calibrations like you've done before. You're making a series of standard dilutions. And by measuring the absorbance of those standards of known concentrations, you can make a calibration curve. This time, we're also measuring a reagent blank. Okay, so this time the reagent blank is because in this experiment, we're adding excess of all of those reactants that are needed to form this blue coordination complex. And we don't know how those excess reagents might be affecting our measurements. So we're gonna have those reagents plus DI is a reagent blank that might have some absorbance. And from there, we can make a working curve, which means we're going to take that amount of absorption that we get from the reagent blank solution and subtract it from all of the other values, because we know that that much of the absorbance is contributed by our reagents and aren't because of the phosphate itself. Okay, so we'll subtract. Let me show you an example of that. Okay, say when you first did your calibration curve before you converted it into a working curve, your reagent blank would be some point on the y-axis because your concentration of phosphate is 0. You didn't add anything to it. It's just plain DI water. Okay. And then you made a series of standard dilutions, so those have some known concentration, and according to Beer's law, they should have an increasing absorbance with increasing concentration. But now we know that this amount here is due to the reagents that we added, which means we can move this whole point down to the origin. And similarly, we can move that distance, that same distance, for all the other points, shift them all down to fix them. And then we can make a trend line through those new points in the working curve. And you'll notice now that we've corrected our calibration curve with a reagent blank, this first point is at the origin, so we can send the trend line through the origin. And that means that there's no plus b on the y equals mx because the y-intercept is 0. Okay, so this is what's known as a working curve. Like you're familiar with in past experiments now, you'll also be measuring your samples and using this calibration curve or this working curve trend line in order to solve for the concentrations. The only thing to note is that we have both low nutrient and contaminated seawater as our samples here. And low nutrient. Think about what is the concentration of phosphate in seawater. It's actually pretty low. And you need to start thinking about, can our instrument detect the concentrations that low? So in the next experiment, the nitrite experiment, you'll be asked to determine what the limit of detection of this instrument is, and that'll help you answer this question, I guess. But to go ahead and tell you, no, our UV-Vis would not be able to detect the concentration of phosphate in low nutrient seawater, which is just plain old seawater. Limit of detection. So that's why we're also giving you some contaminated seawater. Okay? This, kind of like I described at the beginning, maybe it's contaminated because of runoff that had a lot of fertilizer in it, and so now we have really high concentrations of phosphate in the seawater, and now that means we can detect these levels. Detectable. Okay, just also wanted to mention, as per usual, remember as you're going along to do air propagation. Remember to think about sources of air that might be at play using this instrument or using this method. Again, think about matrix effects. What solution are you making your standard dilutions in? Are those comparable to your samples? Okay, accuracy and precision. Always want to talk about those. Okay, good luck, have fun, we'll see you next time.